Hello, everyone. My name is Olivia Kalloo, and I am a current D3 at Roseman College of Dental Medicine. Today, I will be presenting on e-cigarette effects on oral cells metabolism. As an introduction, I'd like to talk about the difference between e-cigarettes and traditional cigarettes. Both of them serve as nicotine delivery methods. E-cigs, however, do not contain tar or other toxic chemicals found in traditional cigarettes. E-cigarettes have a battery as the heating element, which can lead to inhalation of heavy metals such as nickel, tin, and lead from the battery. There are many different types of e-cigs on the market. The model that we are using in this study is the large size tank device, which requires refills of the vape liquid. There are other models that require cartridges to refill, and you do not have access to the vape liquid itself. Furthermore, there are dangers for children and adolescents for using e-cigarettes. With regards to the vape liquid, children and adults can be poisoned by swallowing, breathing, or absorbing the liquid through their skin or eyes. 50% of calls to poison control centers for e-cigarettes are for kids ages 5 or younger, according to the CDC in 2016. Additionally, there are advertisements for e-cigarette vapors all over the internet and on social media that appeal to young children or disguised as candy, dessert, or fruit flavors. In terms of dental clinical relevance, nicotine is the main ingredient in cigarettes and in electronic cigarettes. Nicotine is the main cause of vasoconstriction. Therefore, when cigarette smokers continue using cigarettes or e-cigarettes, they develop periodontal disease without realizing the progression or extent of the disease. Typically, you would see erythematous gingival tissue. However, due to the vasoconstriction of nicotine, tobacco smokers do not realize the extent of their disease, which leads to more troublesome effects. Furthermore, chronic inflammation induced by a persistent chemical or foreign agents is known to be a risk factor for cancer. Immune response generates metabolic changes released by reactive nitrogen species and by releasing reactive oxygen species, ROS and RNS. Nicotine exacerbates cytotoxic and pro-inflammatory effects, which can lead to DNA damage. In this model, it shows that cigarette smoke or e-cigarette smoke leads to mitochondrial dysfunction due to chronic inflammation or metabolic alterations and increasing in oxidative stress through reactive oxygen species and reactive nitrogen species. Both of these pathways can lead to cancer. Recent findings suggest that e-cigarettes deregulate genes associated with molecular pathways related to the cancer disease process. It is thought that the quantity and how often the individual is exposed to the vapors are factors that will determine the extent of damage in the oral cavity. Our hypothesis is that oral cells exposed to vapors from e-cigarettes will induce mitochondrial dysfunction and eventually lead to cancer. Overall, cell toxicity will be measured using an LDH assay, showing an increase in lactase dehydrogenase release. Secondly, oral cells exposed to vapors from e-cigarettes will induce DNA damage. There will be an increase in histone-associated double-stranded DNA breaks. In terms of the materials used for this study, we will be using a third-generation e-cigarette device, which is the large tank device here, that will be used to create the e-cigarette vapors. The extracts will be generated by bubbling the vapors from the e-cigarette into the cell culture media using this pump system. The oral cells will then be stimulated with differing concentrations of this extract and different time points. Cell viability will be monitored and analyzed for mitochondrial dysfunction as well as DNA damage.
E-cigarette extract will be obtained from nicotine and nicotine-free e-cigarette liquid using the flavors menthol and orange dream. Oral cells were treated at different time points and at different concentrations. We will observe mitochondrial dysfunction by measuring NAD/NADH ratios, measuring LDH cytotoxicity, measuring mitochondrial proteins, and measuring of total ROS. We will also observe DNA damage by measuring H2AX, which is histone associated with double-stranded breaks, and measuring expression of DNA repair factors. So for our results, HGF1 cells were plated in a 96 well plate and allowed to grow for 96 hours with serum and antibiotic-free DMEM media. The cells were exposed to vapor-infused media using two flavors and concentrations, menthol or orange blast, at either 30% or 40% concentration, respectively. After a cytotoxicity assay was performed to measure cell death in response to different concentrations, exposures, and flavors of the e-cigarette vapor. So for our results, the e-cigarette vapor was infused into cell media. The 40% orange dream and menthol flavor blast were the most cytotoxic over a 10-hour period, respectively. The 30% menthol blast and orange dream, both without nicotine, were shown to be the least cytotoxic, respectively. All of the e-cigarette vapor media had some level of cytotoxicity, which increased over the exposure period. So for our discussion, we found that Orange Dream was more cytotoxic than the menthol blast flavor, both at 30 and 40% dilutions, and with or without nicotine. Vapor liquid that contained nicotine was more cytotoxic than the flavor without nicotine. The flavors without nicotine still showed a degree of cytotoxicity, indicating there are substances in the vapor flavor itself that are harmful to the HGF1 cells. So for future studies, we would like to include an understanding the amount of e-cigarette smoke that can induce mitochondrial damage in the cell by looking into NAD/NADH ratio, because this ratio is associated with an increase during cancer progression. Furthermore, we would like to study mitochondrial DNA damage, as nicotine has been shown to affect mitochondrial function, and its effects can lead to DNA damage, apoptosis, and chronic inflammation. E-cigarette toxic agents cause stress in the cells, leading to an inflammatory response. However, because of its toxicity, it seems to affect immune genes. We hope to elucidate a connection between e-cigarette metabolic changes and cancer development that will help to focus immunotherapies to target specific pathways for cancer treatment. In conclusion, e-cigarette vapor has toxic effects to oral mucosal cells. Human gingival fibroblast cells were used to study the effects of toxicity of the e-cigarette vapor with two flavors, menthol and orange dream, with and without nicotine. The higher concentrations of the two flavors at 40% dilution, orange dream and menthol blast with nicotine, were the most cytotoxic to the HGF1 cells. However, cytotoxicity was also shown in the lower concentration vape liquid without nicotine as well. This suggests that the flavors themselves, even without nicotine, are harmful for the HGF1 cells. We would like to thank our advisors, Dr. Freitas and Dr. Awan, for their support, as well as Roseman University for allowing us the opportunity to conduct our research. Thank you.